This lecture is meant to follow the lecture on the cell membrane, and it is an overview of all of the other structures of the cell membrane being the most important part of the cell. It's the only part of the cell that if you put a hole in the membrane, the cell is dead. However, a cell can live without a nucleus, a cell can live without mitochondria, etc. So it's the most important part of the cell. And although many students at the end for an exam say want to match the mitochondria is the powerhouse of the cell, or the nucleus has the chromosomes. Obviously, you want a basic understanding of this. I'll also throw in a bit more just to show how interesting these cell parts are, how much our increases in how profoundly we understand the cells, or appreciating our understanding of the nature of disease, etc. So they'll be a little bit more than just the basic definition types of things, because in our modern age, where our advances in biology are occurring at a faster rate than at any other point in human history, our appreciation of the cell is certainly changing. So first off, the cell is the basic unit of life. Everything that is alive is made of one or more cells. Nothing that doesn't have cells is considered to be truly alive. Now, I'll be focusing on the great complexity of eukaryotic cells in this video. And that's different from the simplest of cells, the prokaryotic cells, the bacteria. Now, that's not to dismiss bacteria. Obviously, an interesting topic in humans is the idea of a microbiome. There's actually more bacterial cells associated with the human body than there are human cells. When you look in the mirror, the number of bacterial cells in that mass outnumbers the human cells in that mass. And so, bacteria, these prokaryotic cells, are certainly very important. And, when we talk about eukaryotes, the mitochondria of human cells seem to be the descendants of bacteria living inside larger cells. And so prokaryotes are fascinating, but I would cover them more in greater detail elsewhere. The eukaryotic cells are typically larger, and they typically have membrane-bound organelles inside, like the nucleus and the mitochondria that bacteria lack. Now, we are not the only eukaryotic cells, we humans or we animals. Plants, fungi, also algae and amoeba and paramecia, they are eukaryotic cells as well. And so thus, when you look at a eukaryotic cell under the microscope, you're struck at how complex it is compared to, say, a prokaryotic cell. So it is larger over average. In general, you can see a large nucleus, the largest organelle of the cell. But then there's also lots of other things going on in the inside. So in this video, in this amoeba, you'll see all of these smaller structures, which would include lysosomes, mitochondria, etc. Also, one of the fascinating things is the cytoskeleton. Unlike bacteria, eukaryotes form things like cilia, true flagella, different from the bacterial structures, but also have a cytoskeleton made of, say, actin that other proteins like myosin can pull on. And so here you see, a cell which is actually moving. In this case, it kind of rebuilds its cytoskeleton in front of bacteria. Organelles make these cells unique. And all eukaryotes would share these features. So when we look at one-cell organisms, they have flagella, like the flagella that you would find, say, on human sperm. You can find cilia, like the cilia which line outward. So eukaryotic cells just more complex. Is the nucleus, and this is where most of the DNA is held. I say most because there's also DNA in mitochondria. And this then told that one single cell how to develop into a human, a multicellular individual with a skeleton, eyes, a liver, blood vessels, etc. So the instructions are here. If you could take the DNA, say a nucleus out of, say, a cheek cell, fool it a little bit to think it's not a cheek cell, and put it into an ovum whose nucleus has been removed, this is a type of cloning. You could actually have a twin. You could have someone who looks pretty much just like you, because the information in one nucleus in one of your cells is the code for you. All right. There's 3 billion nucleotides here. And if you were to stretch out all of the DNA in one cell, it would be about 6 feet in length. And thus all of your DNA, would stretch to the moon and back thousands of times. We keep all of it in this nucleus, which is surrounded by a double membrane, which we call the nuclear membrane, or the nuclear envelope. This nuclear envelope has holes in it, pores, because there are messages which go into the nucleus telling it, oh, you just cut ourselves, we need to divide. Here's a signal because we haven't eaten in a while, and so the things you have to do are different from after you've just eaten. Or, for example, estrogen and testosterone will go through these pores after binding to their receptors and tell the cell, hey, we've started puberty. The messages we want you to send to the cell for the proteins that you make are now different. And so those nuclear pores are where messages can go into the nucleus, and where RNA molecules, as we'll see in a second, can then leave the nucleus. Now, this cell has a cytoskeleton, which I'll be covering in a little bit, but there is also a nucleoskeleton. So just as there are protein fibers composing the cytoskeleton, there's a nuclear skeleton composed of proteins known as lamins. And not only does it give the cell the nucleus its shape, but this then can attach to chromosomes into the nuclear envelope and be involved in, the organization of the chromosomes, the chromatin, the expression of genes. And there are a number of disorders which can be called laminopathies, where a problem with nuclear lamins, maybe a mutation in one of the lamin genes, then contributes to cancer, to diabetes, to not being able to sort chromosomes normally, and leading to abnormal numbers of chromosomes, chromosomal breaks, and then this can be a cause of cancer. So the proteins in the nucleus, which give the nucleus its structure, the nucleoskeleton, they're important and have roles in disease. Now, every time a cell divides, it needs to replicate its DNA, because if it didn't, then once cells started dividing, if they just started taking their DNA and splitting it, then after a while you wouldn't have all the right genes. So each time cells divided, we had to cut our genetic information in half. We'd get to a point where we would have some chromosomes but not others. And that's then a problem, because all cells need the same chromosomes, right? So you have 46 chromosomes in your cheek cells, 2 copies of chromosome 1, 2 copies of chromosome 2, etc. You have 46 chromosomes in your bone cells. You have 46 chromosomes in your heart muscle cells, in your brain cells. And so this nucleus has essentially the same genetic information in virtually all human cells. There are a few exceptions, which I'll get to at the end. So this is where we keep 46 chromosomes, which have the 3 billion nucleotides in the human genome. And our genes are then on the chromosome. So if you make the protein hemoglobin, the instructions on how to make hemoglobin are on one chromosome, while the instructions on how to make, say, a tumor suppressor protein to prevent cancer is on a different chromosome. Every time cells divide, we have to replicate this DNA. And so, when we mention mitosis, information. So if we were to just look at this one, now, if you ever want to use a gene, to say make a protein with the correct amino acid sequence, well, that's a problem, because the essentially have to photocopy the gene and then photocopy to the protein's. So this process of transcription is also a nucleic acid, not DNA, but RNA. So RNA has a different sugar in its nucleotides, so that's ribose, deoxyribose. So and RNA. Where do genes all of this DNA? Where's gene? Most of the gene starts here. You're able to you can convert into RNA and then protein, because there are start sequences. The RNA is made in the nucleus, and then it leaves through those nuclear pores and then reaches the cytoplasm. So this long blue strand, this is representing the RNA molecule which has left the nucleus. And then these structures known as ribosomes form on the RNA. Remember, the RNA then has a copy of the DNA. So the acid is different from the amino acid. Some, for example, are polar. They can interact with water. Some are non-polar, more acidic, in your blood. And so these put them together in one order, you get keratin. If you put them together in a different order, you get hemoglobin. If you put them together in the wrong order, if there's a mutation, you might get a clotting that doesn't work, causing hemophilia, or and so the code is very important to be able to continue without that. It has this code RNA, and that's the cytoplasm in structures known as ribosomes. Ribosomes are where the RNA, which is called messenger RNA, having come from the nucleus copying the DNA, where this will then be read so that amino acids can be put in a precise order. They have molecules and there's a note. Look how complex these ribosomes are. They have big pieces of RNA, and then they have all of these proteins. So, our small subunits have 33 proteins. Our large subunits have 49 proteins, in addition to those large molecules of RNA. Now, just once again, I wanted to, throw out little sentences here and there which give insight into deeper understandings. Those ribosomal proteins, we often don't talk about them, but they do matter. Some are kind of universal, present in all ribosomes, but some are specific, that are specific in different tissue types, or specific promoting the translation of different genes. And so we want to regulate our genes, which ones are turned on, which ones are turned off. And so one of the ways that we do that is by regulating translation. And one of the ways that we regulate translation is by these ribosomal proteins. So there's different kinds of ribosomal proteins, and they are a little more complex, than most people give them credit for. So once again, ribosomes can be floating freely, but a very large quantity of them are attached to a membrane system of the cell known as endoplasmic reticulum. When ribosomes are attached to the outside of the endoplasmic reticulum, it gives the ER a rough appearance when you look at them under, say, an electron microscope, because you have all of these bumps, and the bumps are the ribosomes. So here you see the dark blue membrane system. It's huge, but notice the little white dots. Those are ribosomes. So a lot of RNAs which leave the nucleus, they end up going to the RER, the rough endoplasmic reticulum. And while the ribosomes are on the outside, the proteins which are then made then accumulate in the inside. Right? So once again, the nucleus has the instructions on how to make the protein. The ribosomes, often those are proteins, so the RNAs leave the pores, and the proteins are made at the endoplasmic reticulum. Once again, this is probably beyond an introductory understanding, but not all proteins are made in the RER. There are actually signal sequences once the mRNA starts to be translated into a short little protein. This then binds a signal recognition particle that migrates this whole structure to the RER, where then the translation will continue. And so, that's just one of the fascinating things. Your cells are really organized. And so certain genes are going to be translated in the cytoplasm. Others are then targeted to go to the membrane system known as the RER using this mechanism. So there's a lot more control and regulation than is immediately apparent. Now, the RER has lots of advantages. Why would you want, proteins to go to the RER instead of the cytosol? Well, probably lots of reasons, but two big ones. One is this is a very important area for controlling the three-dimensional shape of a protein. Shape is very important, and the RER can have a different pH than the cytosol. And that's important because some of the bonds forming which control the shapes of proteins, they will occur at some pHs rather than others. And so if you want a protein to fold a specific way, many of them would not fold correctly in the cytosol. They would only fold correctly in the endoplasmic reticulum, in part because the pH is different. So things like disulfide bonds, which help to control the shape of proteins, are formed here. And then also, once you have the protein in the middle of the RER, what's called the lumen, the space in the middle, then you can ship it because the RER is so big. You can move it around the cell, all right? And so, it can aid in transport. Now, there's a lot more in the way of membranes. And so let me just kind of, mention a bunch with very quick definitions. There is endoplasmic reticulum that doesn't have ribosomes. And because it doesn't have ribosomes, this is then what is called smooth endoplasmic reticulum. It often has a different shape. Instead of looking like, these flat sheets, it also looks like, rounder tubes. And there are proteins which can control the shape that the endoplasmic reticulum takes on. And so this is not a random event. One of the things, I'm just going to mention a bunch of them, a lot of textbooks will say there's rough endoplasmic reticulum, proteins are made here, there's smooth endoplasmic reticulum, but then there's so many other membrane systems as well. Oh, I'm sorry, we left one video on SER, we'll get back to it. So in addition to the rough endoplasmic reticulum, there are exit sites, so endoplasmic reticulum exit sites, or ERES, where now we form little membrane bags, which can say ship a protein from the RER to the SER, or to any of these other areas. So there's a whole bunch of membranes. So here's a membrane system, there's a membrane system, there's a membrane system, there's a membrane system. And then there can be transport from one to the other. Before a protein would say go to the Golgi, there's what's known, so here's the Golgi here, which we'll get to, but there's an ER to Golgi intermediate compartment. And so, these proteins, they're now in a membrane system, but you can form these little membrane bags and then ship them, from one place to another. And so, notice there's, transport going back and forth. There's also then an area where protein quality control can be measured. So a membrane system known as the ER quality control, where protein then it could be because they have that they are cancerous and that they should you need to have ribosomes. So the SER can have then so in addition to the RER, which is where proteins are made, the protein is made there because there are ribosomes on the surface. Then some of these can be then sent to these tubes, the SER. And SER can have a lot of different functions, but for example, that could be where non-protein molecules are made. Cells need to make cholesterol. Cells need to make phospholipids. Cells need to make a number of things. And so enzymes, which were made in the RER, could then be shipped to the tubular SER that lack the ribosomes. And this could then be an area where non-protein molecules are made. There's a prominent set of membranes known as the Golgi, which is where final touches can be put on proteins. So maybe final parts to their structure or their folding. Also, sugar groups can be attached. You know, many proteins are actually glycoproteins, where they're not just a protein, they have a sugar group attached. Some proteins, when they're made in the RER, they don't work because they need to have a couple of amino acids cut off of one end before they are activated and become functional. And so from the RER, many vesicles, many membrane bags can form, which go then to that intermediate compartment, which then go to the Golgi. And as these membranes are then processed, then these proteins are, you know, their folding is worked on, sugar groups may be added, you know, they can be activated, and then the Golgi will ultimately then ship these proteins off to where they need to go. So it's kind of like a packaging center where the final touches are put on proteins to make them ready, and then they are shipped off. So some of these proteins may be bound to be secreted from the cell. Some may be bound to be on the cell membrane as a cell membrane protein. Some may be going to the mitochondria or elsewhere. And so once RER make proteins, these proteins can be shipped to an intermediate compartment, then to the Golgi. The Golgi puts the final touches on them, and they can be sent throughout the cell. So once again, the cell is very complex. And if you truly want to understand, you know, disease, you know, and the functioning of the human body beyond a certain level, you know, then you need to pursue, you know, the cell rather than, you know, just, you know, the one sentence definition, which, you know, for a multiple choice test, which, you know, is lysosomes. So if you have a membrane bag that has digestive enzymes in an acidic juice, that's a lysosome. Lysosomes are made here at the Golgi. The Golgi put digestive enzymes and proton pumps into a bag. This then becomes the lysosome. So a big part of a cell is the proteins that it makes. And to explain that, I talked about the roles of the nucleus, the nuclear pores, the ribosomes, the RER, the SER, the Golgi in doing the final packaging, and then also mentioning there are additional, you know, membrane compartments in the cell as well. The second largest part of the cell is the mitochondria. So the nucleus is the largest organelle. Mitochondria are the second largest organelle. And cells can have lots and lots of mitochondria. Mitochondria have an outer membrane and an inner membrane, which is highly folded, forming these shells known as cristae. And so here we see, you know, the mitochondria close up. And mitochondria are fascinating for lots of reasons. One is they actually have a little bit of DNA. So when if I say humans have 46 chromosomes, it's actually a lie. They have 46 in their nucleus, but each mitochondria has its own chromosome. Now it's small and it's circular like a bacterial chromosome, not like a human chromosome, but it does encode genes. Some people do have genetic diseases or even cancer, not because of a mutation in their and not because of a mutation in their nuclear genes, but because of a mutation in their mitochondrial genes. We get our mitochondria from our mothers. Sperm really don't carry mitochondria, and if they do, they tend to stay on the outside of the cell during fertilization. So here's a mitochondria and here's a mitochondrion for the embryo. All of them, the mitochondria carried since our father's sperm did not contribute. So here, once again, just a depiction of the mitochondria with its outer membrane inner membrane forming these folds known as cristae of the nucleus. Here are the cells of a turtle, and all the little dark dots are mitochondria. So the largest organelle of the cell is the nucleus, as you can see. The mitochondria are the second largest organelle, and as you can see in these cells, that cells have lots and lots of them. Red blood cells, interestingly, not only eject the nucleus, but also eject their mitochondria. So not all cells have mitochondria, but many cells, like a huge skeletal muscle cell, can have thousands and thousands of them. Lots of things happen at the mitochondria. The main thing is this is where we create energy from the food we eat. So this is, part of other lectures, say lectures, in the muscular system or on the metabolism. But when you eat food, all right, you break it into, pieces, and, perhaps glucose you break in half. And then you take those halves to the mitochondria, where you have first the Krebs cycle, where the carbon in the food you eat is converted to carbon dioxide. But then you also have the electron transport system move the hydrogen from your food to that inner membrane. And it's at this inner membrane that we create ATP, thanks to the hydrogen from the food in your last meal. And this is the ATP that essentially you run on. So most of the energy you have ever used in your life has been generated at the mitochondria from the food that you eat. Once again, that's an involved process. Cellular respiration can be discussed later, but that occurs at the mitochondria. Once again, it has a small chromosome with genes for 13 proteins, 22 tRNAs, and 2 rRNAs, so there are genes here. And then one other thing, also part of another lecture, is that one of the ways we help protect against cancer is that we try to kill cells which are behaving abnormally. Our own cells can self-destruct, and the mitochondria play a role in this. And so mitochondria can release proteins which help kill the cell, and that's actually something that we can do on purpose, once again, in something known as programmed cell death or apoptosis that we can discuss with cancer when we're afraid that cells are behaving abnormally. Okay. Before I get to the final topic of the cytoskeleton, just to wrap up then, the cell's a big place, and it's very highly organized. And there's all of these different sections. And if you were to ask, what, part of the cell is doing a specific function, where is a specific protein found? There are some proteins which are only found on the cell membrane, some proteins which function in the nucleus, say by sticking to DNA, some proteins which function in the mitochondria, some which function in lysosomes. Just like the body does that role for the mitochondria, etc. The next one, the lysosomes, which once again have a role in digestion, where if we can put digestive enzymes and put in a bag, this then becomes something which can, say, fuse to a vesicle with bacteria. But we can use lysosomes for lots of things, like, we have to, if we have to dissolve bone, and cells known as osteoblasts, other membranes as well. So just mentioning this is, perhaps a deeper understanding of the ER mitochondria. And the parts which contact mitochondria, they're special. So a special part of the cell are what are called mitochondria-associated membranes. So we have an RAR, we have an SER, we have a Golgi, but the membranes which touch RAR, the special roles. And so that would be hard to understand. There are other types of vesicles, some which can help cells destroy themselves or destroy materials in the cells. If materials are being recycled, called autophagosomes. And so, very often an introductory text, will list, the nucleus does this, the mitochondria does this, and then you go into the next section. But cells actually have more regions and very highly specialized functions, which we are just starting to understand. And just as there are proteins which can move, or sequences which move proteins to, say, the RAR, there are coding sequences which are to this compartment or that compartment. So the inside of your cell is very highly organized. To describe is the cytoskeleton. Um, Now, I have a lot of videos that go into this in in more detail than I'll present here. This is just an overview, but if those videos are of interest. Why do we have a specific shape? Why do we look the way we do? The answer is, well, because we have a skeleton with that specific shape. And if you were to take the bones out of a person, if you were to remove the skeleton, well, then we would be, just kind of this, blob on the floor. And in the same way, cells have a cytoskeleton, which gives them shape. You have a specific shape because of your skeleton. Cells have a specific shape because of their skeleton. Your bony skeleton is made of bone, which has proteins and then hard calcium salts. The cell's skeleton is just made of proteins. So, our, skeleton has protein in it, like collagen. And there are proteins which make up the cell's cytoskeleton. Now, there are a variety of proteins here, and they vary in the cell types that they are most found in. They vary in their size. The narrow ones are called actin. This includes a web which is just deep to the cell membrane, and then also actin filaments which are in the nucleus. And so, once again, it's not random, it's very highly organized, but thin filaments of actin make up the cytoskeleton. From hollow tubes, nucleus, which can be does give a cell move along tracks, and so this helps give the cell, an organization that all of these organelles can then make use of. There's different types of actin, and so cells vary how much actin is in them. So you can see a typical cell 1 to 5%, but could be 20% in muscle. And in muscle here, the actin is organized, as you can discuss later, into these filaments that then another protein called myosin pulls on. And when a muscle contracts, it's because myosin here in the middle in red pulls on actin, and that's what causes the muscle the sarcomere of the muscle to contract. And so talking about muscle contraction, we'll then have to revisit the topic of actin. Earlier in the video on when I talked about eukaryotic cells, you noticed that this eukaryotic cell was moving. And the way that it does was it was taking apart its actin skeleton in one part of the cell and then kind of rebuilding it in another. It was kind of, flowing into its skeleton. And our white blood cells move with this type of amoeboid function. So you can take apart the cytoskeleton at one part of the cell and then rebuild it at another part of the cell. So not only do amoebas move in that way, but also white blood cells move with this amoeboid function. And so there are, that's that amoeboid motion I just described. And so we have various types of actin that can vary throughout our body. So we have a cytoskeleton, we have a nucleoskeleton, and the two are actually linked together. There's something actually just referred to as the link complex with specific proteins, which then span the nuclear envelope. And so we can organize things. So for example, when chromosomes are moved, they can be moved because they can be attached to parts of this link skeleton, which is attached to the cytoskeleton. The microtubules, which can form those tracks, which I had mentioned earlier, can also then cluster together to form larger structures such as cilia, flagella, and centrioles. So let me just say a few words about these. So microtubules are these hollow tubes, and they can form a specific structure where we have a number of doublets of microtubules and then two central microtubules which are present singly. And so this organization is what we find in cilia, in flagella, and in centrioles. And so those microtubules cannot just only exist as single tubes making complex structures. I'll get back to that in a minute. And kinesin proteins, which allow things to actually form microtubules, as we'll see. Microtubules can form these larger structures. They can form short cylinders known as centrioles, which have roles in cell division, which will be discussed later. The centrioles have a slightly different structure than the cilia and flagella because they are made of triplets as opposed to the doublets we find. So if cells have too many centrioles or the centrioles aren't functioning normally, this can have repercussions like cancer, and this can be mentioned again in the topic of cell division. Cilia and flagella have a basal body that look like centrioles. So it looks like there's centrioles at the base of cilia and flagella, and these structures then project from the cell. If cells have lots of short hair-like structures, these are called cilia. So here you see the cilia of a paramecium. Our human cells can have this. So we have cilia lining our trachea that helps to sweep mucus, which has bound things on our delicate lungs, but then that also results in the cilia pushing the air towards cells have one or several large filaments, which are then called flagella. In humans, the only cells which have flagella are the sperm. The flagella of sperm and the cilia of, say, the trachea or the oviduct. They have the exact same structure. The difference is cilia, there's lots of them. Here's a one-celled organism that has a flagellum. When cilia are present, there are lots of them as opposed to one single flagellum. Now, the more we understand cells, the more surprises there are. So for example, it turns out that most, maybe virtually all cells have at least one cilium. All right, so we notice the cilia on the trachea, etc. But most cells have one single cilium. Once again, at the base of the cilium is a basal body that looks like two centrioles. And it turns out it's important. All right, the way I think of it is maybe it's like a compass needle in a sense that because there's this one structure, it helps cells like orient themselves or realize where they are. But this is so important that disorders which affect this cilia, now we know underlie a number of diseases. So there are a number of what are called ciliopathies, which involve abnormalities of this one primary cilium. And so cilia aren't just this, exceptional structure which, is present, in a few tissues. It can underlie, and so almost all of our cells have a primary cilium, which seems to have an important role in embryonic development and then just in normal metabolism. And there are ciliopathies, diseases, which are linked to abnormalities of this cilia. So once again, just an interesting factor. Some ciliopathies, also abnormal numbers of centrioles can be linked to cancer. And these cilia in some cells then get to be specialized. So there are some things that we say, oh, look, here's a weird cell in the eye which is perceiving light. Well, that can actually be a modification of this primary cilium. So primary cilia can be hormones, receptors cilia. And so that works with just a little addition and thing that are associated with microtubules and allow movement along a microtubule. So if you think of a microtubule like a pathway in your cell, and moving a chromosome, say during cell division along a specific and so this wraps up my kind of synthesis of the parts of a cell. And very often at this point in the class, I say we've talked about the average cell, and knowing that in an anatomy class, the class on tissue, the chapter on tissues is coming, I'll ask my class, well, where in the body would you find an average cell? And the point that I'd like to end with then is you don't. The average cell, it's a myth. All right, so that I've said, oh, cells have a nucleus, cell have this, cell have this. Well, no, they vary. All of these structures, they can vary in cells. And that's interesting because as we transition from cells in the average cell into human tissues, we can then say, well, what makes one tissue, one organ different? Very often it's variations in the cells. So for example, liver cells are just weird. Some have more than one nucleus. Inside their nucleus, instead of having 2 copies of each chromosome, some have 4 copies of each chromosome or 8 copies of each chromosome. They can be polyploid. And so therefore, I'd said, this is how the nucleus is in a cell. In a sense, that wasn't entirely true because there are exceptions. Not all human cells are examples of that. So most cells in the nucleus. That's what's known as being diploid, having 2 copies of each chromosome. Not only are liver cells an exception, but there is a cell in the bone marrow called a megakaryocyte, or they can get really big and be called metamegakaryocytes. They can have 8, 6, 32, or even 64 copies of chromosomes. So each of the more sets of chromosomes, the more platelets that each of these will make. That then is not all cells. Some of your cells have lots of nuclei. Inside one cell, there's multiple nuclei. And one of the reasons that when you have class, which white cell use, one of them must be together. Has 2 copies of each chromosome. It is diploid. The obvious exception to that would be the sex cells, the one of each chromosome that have to stay in one cell as opposed to only when lots of cells have skeletal muscles, absolutely huge, hundreds of nuclei, and they can be a foot long. You can see them without a microscope. They're so wide. How did they get to be cells called myoblasts in development? And so there are multiple human cells that have more than one nucleus, which have more than of chromosomes for a number of reasons, and I go through some of them there. There are cells which lack nuclei, the red blood cells, mitochondria, as so as a rule. Cells lose their cells and thus are again, like nerve cells. That might be a way we protect against cancer in some cases. We prevent cells from ever being able to move out they need for that. Of estrogen and testosterone, which are not proteins, they're steroids. You would have more SER, while say, the intestinal cells, which make more digestive enzymes, you would have more RER. And so I have just a series of videos here. You're welcome to look in greater detail, which kind of offer the transition. Now that we've talked about the average cell, in your body, there are no average cells. Each cell is specialized. And so thus some of the structures, which I had mentioned, they vary. So the nucleus we talked about. But once again, red blood cells eject their nuclei, and there are other variations of that. I talked about the RER, the SER, the Golgi. Well, cells vary in that as one specializes. Some cells have cilia, sperm have flagella, some cells have lost their centrioles. Some cells have modified that primary cilium to be sensory, to perceive light in the eye or odorants in the nose, for example. And so I think this is a nice transition between this chapter on the cell, showing that now throughout the body, different cells vary in these structures. And this then introduces the next chapter on tissues.